Hello everyone, this is your Daily Dose of Biology. My name is Benjamin XU from 5th period, and today I'd like to tell you about what caused my explosive diarrhea last weekend, Part 5, Cellular Respiration. Last time we reviewed my failing intestinal system, and today we will go over a much smaller structure, the mitochondria. The mitochondria is an organelle that is often referred to as the
powerhouse of the cell.
But it's a little bit more complicated than that. In fact, the mitochondria is a membrane-bound organelle that tirelessly works to provide the body and cells with ATP, which is a widely available source of usable chemical energy. How is this ATP made, you ask? Well, first, we need 30 to 32 ADP and inorganic phosphate, and then we combine glucose and 6 O2 molecules to produce 30 to 32 ATP, and 6. The ADP stands for adenosine diphosphate, which means it has 2 phosphate groups, so it's rephosphorylated with another inorganic phosphate to make ATP, or adenosine triphosphate. However, some kinetic interrupt your regularly scheduled programming for a special on the cellular. Hello, everyone. Welcome to the Today 12:09 a.m. Weather Politics Educational Palooza Hour. 3.0 Good Morning Sunny Afternoon Nationalist Propaganda Show for kids. Today's special is about a very special type of food that I ate, and it potentially gave me indigestion: popcorn. Popcorn is a food known for having high amounts of carbohydrates, and given that it might have caused my indigestion, I don't know how happy that made me feel when I ate it, but
the important part is that the carbohydrates extracted from popcorn are broken down into individual monosaccharide units of glucose via physical or chemical means such as chewing or enzyme activity in the saliva or stomach acid.
These glucose molecules can then be transported via the bloodstream to all the cells of the body, which can then go through cellular respiration to produce more ATP.
A specific cell in the body that uses a lot of ATP to function is the muscle cell. It uses ATP to perform contractions, which allows the cell to complete its purpose of moving around body parts. These contractions and movement allow me to run to the refrigerator whenever I want. Another ingredient we need for cellular respiration is oxygen. Oxygen comes from the air, and when someone breathes it in, it is then pumped into the bloodstream, and just like glucose is sent to every single cell in the body, which will then utilize it for cellular respiration to produce ATP.
Hello everyone, and welcome to Cephalopod Game, copyright 2021. Okay guys, your final task is to answer this last question. What? What is this even supposed to mean? What is a multicellular organism? What are you? Oh, I know what this is. The answer is B. I'm done! How is he already done? This is ridiculous. Okay, everybody, time is up. Turn in your exams. I'm not even done. Okay, after tallying the results, we found that number 432 is the winner of Cephalopod Game.
In fact, everyone is a multicellular organism that is made up of eukaryotic cells. These cells are much more efficient than their prokaryotic brethren because, first off, they act as a small container, which increases the risk of the chances of interaction between enzymes and their substrates. Additionally, they can maximize surface area within the inner membrane with like these folds and humps called cristae. So imagine this is like this sheet of paper is the outer membrane. The inner membrane is going to be looking more like like this, and these folds are like little spikes. These folds are there to maximize surface area and to increase the rate, to increase the volume or concentration of enzymes within the mitochondria. Third, it lines enzymes up in this like assembly line thing, which means that substrates that go into like an enzyme chain go into one enzyme, and then it goes out of the enzyme and then into another enzyme. So that also increases productivity and optimizes reaction rate. Next, it's also like a closed system and can control the pH and salinity and other variables within the membrane, which can optimize the enzymes. Finally, the membrane is selectively permeable, so the mitochondria can choose which substances to let in or out, which also maximizes the efficiency of the enzyme, so it doesn't have to interact with any inhibitors. Moving on to the wider cellular environment, the mitochondria is situated in a goo-like substance known as the cytoplasm, which houses other organelles such as the nucleus, the endoplasmic reticulum, and the Golgi body. Most importantly is the nucleus, which contains all the DNA for the cell and enables it to do its basic functions such as coding proteins and transcribing DNA and RNA. Additionally, the cytoplasm also contains random enzymes and substrates floating around. The entire cell is also covered by a phospholipid bilayer, which is also semi-permeable, just like the mitochondrial membrane. It's fluid mosaic as well. Here we have the overall mitochondrial structure, which has all of the important pieces and parts of the mitochondria, which include first off, the outer membrane, which is the large shell encompassing the entire structure. And in it, there are these small holes called porins, which let specific compounds to enter the mitochondria. And second, we have the inner membrane, which is the membrane that has a lot of folds and bends in it. These folds are called cristae, which, as I mentioned before, allow for optimized enzyme placement. And then next, so the space between the two membranes is called the outer compartment or the intermembrane space. And within the inner membrane is the matrix, which is where a lot of enzymes, granules, ribosomes, and DNA reside.
We can now move on to the processes of cellular respiration. First, glycolysis.
Moving on with the environment for glycolysis, glycolysis takes place in the cytoplasm, where there are many different enzymes bouncing around, waiting for interactions with their respective substrates. There are two different types of enzymes needed for this process, namely kinase and dehydrogenase enzymes. Additionally, this process is completely anaerobic and does not require the presence of molecular oxygen to function. We can now move into the energy in phase of glycolysis. First, glucose, which is a 6-carbon sugar, interacts with hexokinase, which attaches a phosphate molecule that comes from ATP. And then the glucose with the phosphate moves on to interact with isomerase for it to become fructose, and then interacts with phosphofructokinase to become fructose with 2 phosphate molecules on it. It then splits into 2 3-carbon sugars, or glyceraldehyde 3-phosphate. In the second stage of glycolysis, which is the energy out phase, the 2 G3Ps go through dehydrogenase and 2 kinases to become 2 pyruvate molecules. The kinases also phosphorylate ADP molecules into ATP via substrate-level phosphorylation, and the dehydrogenase converts NAD+ into NADH. Overall, glycolysis has a net gain of 2 ATP, 2 NADH, and 2 pyruvate molecules. After glycolysis, we can then move to pyruvate entry. When pyruvate tries to enter the matrix, it has to go through a pyruvate entry complex, where a decarboxylase will take off carboxyl groups to form 2 CO2 molecules. Additionally, 2 dehydrogenase molecules will take off hydrogens to convert 2 NAD+ into 2 NADH. And finally, an enzyme called CoA, quote-unquote, smushase, will create 2 acetyl-CoA molecules, and all of these products will fly into the matrix as a result. The pyruvate entry has a net gain of 2 acetyl-CoA, 2 carbon dioxide, and 2 NADH molecules. It is important to note that glycolysis and pyruvate entry are necessary because both molecules are too large to enter the phospholipid bilayer.
Moving on to the Krebs cycle, it occurs in the matrix, and within the matrix is just a bunch of different enzymes bouncing around in a small container, which increases the chance of interactions between molecules and enzymes and their substrates. The Krebs cycle, or the citric acid cycle, is called a cycle because the end product of the cycle, or Oxaloacetate or oxaloacetic acid is also a reactant for the beginning of the cycle. This is also kind of like a clock, because after the hour hand reaches 12, it just cycles back to 1.
As the diagram shows, at 12 o'clock, or C6, 2-acetyl-CoA will bind to oxaloacetate and will then go through a dehydrogenase, which will rip off a hydrogen to convert 2NAD plus into 2NADH. And then it'll go through two decarboxylases, which will produce a total of 4 CO2. It'll then go through another dehydrogenase to make 2 more NADH, and then it'll go through a kinase at around the 6 o'clock position to phosphorylate 2 ATP from 2 ADP via substrate level phosphorylation. It'll then go through another dehydrogenase. However, this gives hydrogens to another protein known as FAD, which is kind of like NAD, but it is a little more electronegative. And then finally, it goes through another dehydrogenase to make 2 more NADH until it produces oxaloacetate once again.
The overall net gain in the Krebs cycle is 2 ATP via substrate level phosphorylation, 2 FADH2 via the dehydrogenase, and 6 NADH molecules.
Moving on to the electron transport chain, the 5 enzymes on this chain is the NADH dehydrogenase, succinate Q oxidoreductase, the BC1 complex, cytochrome, and the cytochrome oxidase complex. NADH and FADH2 will float around the matrix until it interacts with the electron transport chain on the inner membrane. However, only the larger enzyme complexes, such as NADH dehydrogenase, the BC1 or cytochrome oxidase, will be able to pump hydrogen ions. Each enzyme is more electronegative than the last, which is why it's able to pass along electrons down the chain. However, the final electron acceptor is molecular oxygen, which is the most electronegative of them all. The 2 electrons will be accepted by oxygen, or only 1/2 of oxygen, which will split apart from the other oxygen, giving it a charge of -2, which will attract random hydrogen ions floating around to create H2O, otherwise known as water. When NADH goes into the electron transport chain, it goes through all 5 enzymes and therefore pumps 10 hydrogen ions. However, when FADH2 goes into the chain, it's more electronegative than NADH and therefore skips the first enzyme and only pumps 6 hydrogen ions. A gradient is the difference in concentration of 2 sides, and in the mitochondria, this is the difference of hydrogen ions between the outer compartment and the inner matrix. The outer compartment has a much higher concentration of hydrogen ions.
Since the phospholipid bilayer is also mostly nonpolar, hydrogen ions are also not able to cross the bilayer.
The energy conversions of chemiosmosis start when the electrons are used to pump the protons out into the outer compartment, which is mechanical energy, which is then converted into potential energy because these protons are being added to the gradient. This gradient is then converted into mechanical energy because it's used to spin the knob of the ATP synthase, or the so-called lollipop of life. Pretend this is a lollipop, it's gonna spin. And then this mechanical energy, the spinning, smashes ADP and the inorganic phosphate together to create ATP via oxidative phosphorylation. It's called oxidative phosphorylation because oxygen is at the end of the electron transport chain, is what drives the electrons to pump the protons and then power the spinning of the knob in the end. Throughout the process of cellular respiration, around 8 NADH is produced, as well as 2 FADH2 molecules, which and once it goes through the ETC, amounts to a grand total of 23 ATP molecules. 4 more ATP molecules are produced via substrate level phosphorylation, and the remaining 3 to 5 depend on the 2 NADH molecules that are produced during glycolysis. As shown by the diagram, the 2 shaded circles are called lazy Susans, and the NADH molecules can enter either one. If it enters the first one, it goes through all 5 enzymes and produces 5 ATP. However, if it goes through the second one, it only makes 3 ATP, and that contributes to the differences, and that's why it's 30 to 32. ATP molecules in muscle cells are usually used for contractions, which help the cell and other cells to move around body parts like this. When the body runs out of oxygen, the electron transport chain becomes backed up with electrons and therefore won't take any from NADH or FADH2, even though they'll still be produced. Therefore, NADH and FADH2 will continue to accumulate, and then we will run out of NAD plus and Because of this, the only thing that can happen now is glycolysis because it is completely anaerobic and does not require oxygen. However, 2 ATP molecules are used to phosphorylate hexokinase and phosphofructokinase, and that leads to an even bigger loss in energy. Luckily, the body can use lactic acid fermentation, which uses a dehydrogenase to strip the hydrogen from NADH, turning it back into NAD+. This is good because the NAD+ can then go into the second part of glycolysis and then produce a net gain of 2 ATP. As a result of lactic acid fermentation, lactic acid will eventually build up in the muscle cells, and since it's an acid, it can change the pH of the cells, which causes soreness. The products of a fungus fermenting pyruvate, however, results in the production of ethyl alcohol.
Thank you for watching.